All right, back to you, Pete, to introduce the next speaker.
Thank you, Julia. And I mean, the scientist artists here are blowing my mind. And so I really appreciate Dr. Tall's work. And I'm excited now to introduce our final speaker for this morning. It is Dr. Melissa Duhaime. Dr. Duhaime is an assistant professor of ecology and evolutionary biology at the University of Michigan. Her laboratory studies microbial interactions in the ocean and in lakes on particles to better understand their consequences for ecosystem function and environmental health. In the ocean, Dr. Duhaime's lab studies how virus-microbe interactions influence the fate of marine snow particles, and in lakes, they investigate how microplastic biofilm activity influences the transport and fate of plastic pollution, as well as carbon and nitrogen cycling. Now, I say I've never had the pleasure of working alongside Dr. Duhaime, but her work is making waves, and I just love all the work that they're up to. So I hope you'll pardon the pun. But it is also cool, if I may say so, to see Dr. Duhaime and others have such a neat sort of presence in their sort of outreach and public-facing work. And she's also got a great Twitter handle, Envirophage. I mean, that's pretty cool. But I am really excited to hear her science, and I'm going to turn it over now to Dr. Duhaime to share with you all the great work that she and her lab members have been up to. So Dr. Duhaime, the screen is yours.
Thank you, Pete. Right. Bear with me as I go to sorry, tiny screen.
No worries, it looks good.
I think we're ready to go. So yeah, thank you for the introduction. And indeed, we do different types of outreach, but the heart of what I'll focus on today is the research. So there are two arms of the work that we do in our lab, and today I'll talk about the work that we do looking at the microbe and virus interactions and what that might mean for ecosystem scale processes. So we'll look at the virus's guide to infecting bacteria, how to reprogram a host to meet your replication needs. So we'll start with the Earth is our major ecosystem. And we've come to appreciate that microbes rule the world. I think many of you heard in several seminars and readings that the microbes are driving major redox reactions that are essential for life and different biogeochemical cycles. And also, as we look more closely at different classes of holobionts, we're beginning to appreciate the extent to which animals and plants are essentially biomolecular networks that are composed of the hosts and the microbes. So I think more and more we're becoming familiar and comfortable with this idea that microbes rule the world. But what we think about in our lab is the fact that viruses are ruling the microbes when they interact. And the rest of the talk will be an inquiry into how and why that ruling of the microbes happens and what the implications might be. also a general framework for how we're thinking about this. So these are some of the first studies that we've been doing in my lab group, and I think have become pretty foundational in the things that we are interested in or guiding us to our future interests. So we know that, due to, or as a result of decades now of research looking at viruses, first it was like, wow, viruses are there in particular ocean systems. Most of this talk will be referencing. And then it was, wow, there are a lot of viruses there. So we know viruses are incredibly abundant in most habitats that we have looked in. In fact, in most habitats, they outnumber other microbes, sometimes by several orders of magnitude. So that could mean that in every drop of seawater, there could be tens of millions of viruses or more. So this is a big number, staggering, astronomical numbers. I think something on the order of 10 to 30 viruses on the planet has been estimated, and that's really, I have no way to really appreciate what that scale means. It's a big number. And that number has inspired researchers for decades now to ask questions, kind of following up on that observation of numbers, such as why are the viruses there? Why are there so many? And then what are the population and community and ecosystem level consequences of those viruses where they're found? So really, when you put that together, this notion of relationships, it implicitly invokes the principles of viral ecology. And so in other words, thinking about how viruses interact with microbes in their communities, as well as with the environment in which they are found. So these three entities, knowledge of the mechanisms that underlie the interactions between virus, microbe, and environment, I think really underpins our ability to determine whether it's necessary to, for instance, include knowledge of viruses and their impacts in ecosystem scale models, the way that folks are striving to do and succeeding in doing with their microbial hosts. If you think back to the introduction, microbes rule the world, but viruses rule the microbes. How much of that ruling of the microbes do we need to know in a qualitative and quantitative sense in order to best model ecosystem processes? So that's at the heart of the work that I'll show the beginning of today. So if we tease apart, I can give a few more examples of what these interactions actually look like. So between microbes and viruses, you have interactions, so between viruses and other organisms, such as microbes, that could involve horizontal gene transport for the replication process itself, which results in lysis. So that's obviously an effect of viruses on microbes, diversification of both the viral and host populations. Lysis generates lysis products, and it's been estimated that in some habitats, the lysates of a viral infection can serve as food, comprising up to 50% of the diet of heterotrophs that are found in that system. And viruses, while they're infecting their hosts, are also known to metabolically reprogram their host. And I'll talk more about that in detail. And these other interactions between the virus and the environment itself includes processes that influence the abundance and the distribution of viruses, for instance. There are abiotic processes that influence the abundance and distribution of viruses, such as photodegradation and different hydrodynamic transport processes. But then the impact of viruses, so this is free viruses, virions we call them, impact of those viruses on the environment include things like the ecological stoichiometry of the system. So virus particles themselves are reservoirs of carbon and nitrogen and phosphorus, and have also been implicated in kind of how iron moves through marine systems too. And this kind of final interaction, I think we know most well about between microbes and environments. elemental cycles, environmental engineering that microbes are able to do to transform and create their own microhabitats. And the environment provides energy and substrates and really defines the limits of life. But what happens then in the specific instance where that microbe is the microbial host? Well, an infection ensues. And so and this new entity is created that's been termed a virocell. So a microbial host, well, the virocell itself is a cell unit whose function is no longer to produce two daughter cells, but rather to produce viruses or free viruses we call virions to propagate viral genes. And this was a notion introduced 10 years ago now by Patrick Forterre that has really been transformative in the way that I think we think about the possible implications that viruses could have on the environment. So a microbial host that's infected by a virus is distinct from an uninfected microbe. So this new cell, the virocell, is then, as a result, a combination of both a host and a viral genome, so a new novel genotype, and it therefore also has new resource needs, metabolic functions that are possible, so new possible phenotypes that were not there before. And it's really the magnitude of these interactions with, in the end, the environment, so virocell environment interactions that has, I guess, led us to come up with some new thoughts. So virocells interact with their environments in a few ways that we are interested in exploring. So they themselves, the virocells, are organic particles with their own elemental stoichiometry that's distinct from that of a free virus or a host. But they are also still using energy. This reminds me to our first talk today about the concepts of life. So we often also delineate those. What is energy doing? What is carbon doing? Is this thing still alive? What are electron donors and acceptors? And how is that different from that former uninfected host? So just to tie it back to that first talk. So yeah, they use energy to transform organic matter. They also may release and excrete compounds into the environment. And that's another key point for us that we're, I think, far from fully understanding, but the data I'll present today start to bring us towards hypotheses about what that might look like. So I mentioned viruses can reprogram their host metabolism during infection. That means then that in the form of virocells, a substantial portion of microbial cells in any given habitat could be serving the mission of viral replication rather than cellular replication. And this is important in environments like the ocean, where there are estimates that nearly half of the surface microbes are infected with, or may be infected with viruses at any moment. Although, give the asterisk that that is a number that certainly needs better parameterization, I think, but it's a possibility. And so what is the impact of this virocell on its environment? Well, we think of that in terms of an environmental footprint. So I mentioned the possibility for substrate transformation, nutrient uptake and release. And so really recognizing virocells are where this ecosystem action happens, and to better understand the virocell and perhaps build better ecosystem models, there are several questions that arise before first embarking on that pursuit. And I think the first order question is, should viral infection even be included in ecosystem models? So I think we don't know that so far. We've talked about possibilities, possible implications, but it gets complicated very quickly. So if we do include them, for instance, would we need to model each virocell? So that means each distinct virus and microbe interaction. Would we need to model that in every possible environmental situation? Or rather, might we begin to see certain universal features of infection footprint begin to emerge? And what are the bounds of the universality of that? So does that environmental footprint differ from virus cell to virus cell and across environments? Well, those are the types of questions that we're interested in pursuing. And so today I'll focus on the first of a series of experiments that we began, where to better to approach a question, we sought to bound and describe the variation in a virus cell footprint that one might expect given, in this case, varied phage host pairs. So in this case, we tweaked who is the virus infecting a single host. We then followed up on that experiment to look at another parameter to kind of explore the effect space to get at the impact of the virus cell. I won't talk about the results in detail of that experiment. It's unfolding currently. But with these changes, we tweaked these kind of different parameters and then track the molecular responses with multiple omics data types. And so to summarize the first question, how do virus cells differ? Specifically, how do changes in virus cell genotype lead to changes in their phenotype? And by differ, specifically, we think about differences in terms of metabolic reprogramming strategies, work, I'll talk more about that, measures of fitness, viral fitness, and altering their capacity for altering the environment. All right, so first I will introduce you to our model system. Well, now I will introduce you to our model system, which we work with a host called Pseudo-Ultramonas, which I'll abbreviate as PSA in the talk. It's a gamma Protobacterium. We're interested in it in ocean context because it dominates ocean particles, which are important to the biological component of the carbon pump, which brings carbon out of the atmosphere into the deep sea. And it's also been observed through omics data that Pseudo-Ultramonas is the heterotrophic genus that's most correlated with ocean carbon export. So that's one more motivation for why we're attracted to this system in terms of ecosystem impacts. And so we look at PSA by infecting it with two unrelated phages. So one HS2 on the left is Syphovirus, and one HP1 on the right, which is a Protovirus. And importantly, these are very, very fundamentally different viruses. They're genetically distinct. The only area of overlap in their genomes they have is a small domain in a tail spike protein, which we suspect is involved in host recognition, given that they both infect this host. In the genome itself, there's no obvious metabolic genes that might connect it with nutrient metabolism, but there are the canonical phage proteins involved with invasion and replication and assembly and lysis. So those are our players in the system. And one note on another trait that differs between the viruses, when you infect this host, early on we noticed that there are HS2, its burst size is over 60% larger than that of HP1 when infecting the same host. So this to us suggests that HS2 has a higher fitness under the same conditions of infection. Those are all important points to carry on into the experiment. So our experimental set at first addressed the question of how do virus cells differ when we change the virus of the virus host pair. That's what it looked like. In a phosphorus-rich environment, we grew up the PSA host and allowed it to grow, and then we infected it with the two viruses, and in a time-resolved fashion collected transcriptomic data and proteomic data. And by time-resolved, I mean that we captured the discrete stages of infection. We sampled along a time series to try to capture those infection stages for both. So now into the results from the transcriptomics and proteomics data. I'll show two sets of data. The first, remember, collectively, the Vero cell is the viral genome and the host genome, but I'll first show how the host component of the Vero cell genome compared across these three classes, uninfected and the two viruses. And then we'll look specifically at the viral component of the Vero cell genome. So first, in the host, let me see this one. There we go. So overall take home is that the results show that the Vero cells differed dramatically, and that specifically we observed that HP1 here, the red, immediately shut down host transcription. And we were able to observe that over time by looking at the counts of transcripts fitted to a linear effects model. So in the host transcriptome, HP1 immediately shut down host transcription, while the HS2 Vero cell really deviated from the host transcription profile only at the end of infection. And in the host proteome, what we observed was that HP1 translated more host proteins at a faster rate compared to the uninfected. And then at the viral gene component of the Vero cell, we saw that HP1 genes were immediately transcribed, whereas HS2 lagged, and the HP1 proteins were translated at a much faster rate. So what are we seeing here? We see HP1 Vero cell seems to be in some ways working harder for its infection, though we know it's a less fit infection. It produces fewer viable viruses at the end. And so we next wanted to look more at what is the nature of this work. Those are just global perspectives of transcripts and proteins. Was there some Vero cell specific metabolic programming happening? So to answer that, major takeaway is yes. I'll give that off the bat. And what you see on the right are the ways in which kind of elements of sulfur metabolism were reprogrammed differently from the host and also differently from what was observed in HS2. Important for us is that we saw evidence that HP1 Vero cell may be importing sulfate based on the transcription profiles, and that given the overexpression of other genes in this pathway, it likely reduces that sulfate to cysteine. And then moreover, we observed evidence to suggest that that cysteine was being used by the HP1 Vero cell via a specific glyoxylate bypass of TCA. So in some ways, I think of this as HP1 bringing in sulfate to kind of harvest in its garden the cysteine, which then could be consumed or used later for energy. So that was the major observation, observed an HP1 that was not seen in HS2. And so why, so this is more drastic reprogramming. Why does HP1 seem to work harder with this drastic reprogramming? So to approach that question of why and understand the concept of work, we thought more about what the currency of infection might be. And that led us to the path of nucleotides and amino acids, and thinking about who has more and who needs to get more and who has more of what they need to begin with at the start of the infection. So we know that phages, for instance, are master recyclers of nucleotides and amino acids, but as such, they will be constrained by what's already available to them. So we found that HP1 has a greater mismatch. So here on the bottom axis, you see increasing difference between a given phage and its host codon usage. And along the y-axis are the different amino acids encoded by the codons. So you see the cloud of orange spreading all to the right tells us that HP1 has the magnitude of difference in its codon usage is much greater than that of HS2. And so given that translation is the most energy costly task in infection, this led us to the notion of this kind of mismatch burden of evident in the codon usage. And then further evidence to support this is that we found that the HP1 virus-cell overexpresses host tRNAs, so the 43 host tRNAs from the very start of infection, and that's represented by this like warming here. And that's in contrast with the HS2 virus cell that does not overexpress tRNAs, in fact, any. And presumably it's because it's able to use its host's tRNAs. And so this brings us to a notion of the HP1 virus cell needing to compensate early for the burden of its nucleotide and amino acid mismatch by some form of work that we observed in rates of protein synthesis and this taking over the host machinery. And as well, in doing so, influencing the metabolic reprogramming, such as through sulfur import, carbon cycling, and energy and redox elements. And thus, overall, it has the potential for a different environmental footprint of infection. So again, remembering that the only thing that differed here was the virus itself. And so this was really exciting for us because by looking under the hood of this virus-cell activity, we could start to build inferences about what the ecosystem impacts could be of an infection, what underlies variations in that impact, and then possible mechanisms that could explain the differences between the virus cells. And as well as a possible vision for how to connect some of the dimensions of viral ecology. And so what I mean by that, for instance, are life history traits. So we talked about burst size differing between the HP1 virus cell and that of HS2. We then observed that there's this metabolic rewiring happening to meet the energy and resource demands that was evident in both, but more drastic in the HP1 virus cell. And then we also identified the potential for an intracellular energy resource trade-off, whereby increasing the phage host intracellular resource complementarity, you then will decrease the metabolic effort that's needed to carry infection to completion. And based on the inquiry into the codon usage, we were able to offer this phage host genome complementarity as a possible driver of the magnitude and the nature of the ecosystem footprint. So how do virus cells differ? We've shown that the physiology and metabolisms of the virus cells is distinct from that of the uninfected. I'll add, though, that this is largely supporting evidence to suggest and demonstrate the same. That was not a surprise. But what was a surprise was that, that was further supporting the notion that we may need to consider those two infection states separately, infected versus uninfected. But also we observed that, like viral fitness, right, the different burst size on the same host, the nature of the metabolic reprogramming, so that reprogramming itself is a phenotype of a specific phage host pair, and our data support the growing evidence for this. But still, we recognize this doesn't bring us much closer to our first order question of do viruses and these interactions and virus cell broadly need to be included in ecosystem models? And for that's where we will get a little bit more of an idea about the answer of that by changing that environment parameter. So let me see where I didn't include this, but so we start to recognize that there are some universalities in the virus cell response to P, the phosphorus limitation that we impose in our experiment. So there's some universalities. And even early evidence is suggesting that the environment may matter more than the virus cell type. So it's in terms of reprogramming, but what matters are really the ways that that reprogramming interacts with the environment. So for that, we need to, and have explored changes in the exometabolites of the cultures. So now we're looking at the signs of transformation of organic matter in the media to evaluate whether the observed changes in reprogramming, what it means for differences in that environmental footprint of the infection. And so the next task will be to kind of look at the relative impacts of these changes. And I'll say, one of the drawbacks of our current data set is that it's not quantitative. So I see the future of much of this work also pushing the limits of what the omics data can do in terms of absolute quantification. So we can begin to look at fluxes and rates more quantitatively. So I'll leave it at that and give massive hats off and acknowledgements to the lab members and collaborators that have pushed this. And largely that is Christina Howard-Verona from the Sullivan Lab at Ohio State and Morgan Lindbach as a graduate student in our lab at Michigan, who have really spearheaded both of these stories, which are incredibly complicated to piece together between the metabolisms and multiple omics types, and making sense of finally not just describing what the patterns are, but trying to fit it in an ecological framework to answer, like, so what at an ecosystem level. And we're not there yet, but this is how we think about these problems, and this is where we hope to go in the future.
Wonderful. Thank you very much, Dr. Duhaime. That is